The Florida Keys has long been recognized as the birthplace of flats fishing. Beginning soon after World War II, anglers began fishing the Florida Keys for bonefish, tarpon, and permit. In the world of flats fishing, the Florida Keys is hallowed ground. It's the home of most flats fishing world records and is on the bucket list of anglers from around the world. From those beginnings, the flats fishery now occurs throughout the region. Recent economic studies in the Florida Keys, Bahamas, and Belize reveal that the fishery is worth hundreds of millions of dollars annually. Unfortunately, there are many threats to the fishery in the Florida Keys and throughout the region. These threats include habitat loss and degradation from coastal development, dredging, shrimp farms, and other human activities. Declining water quality introduces too many nutrients, as well as pharmaceuticals and other contaminants into coastal waters. Nutrients cause algae blooms that kill seagrasses and damage other habitats. And scars from boat propellers are damaging seagrass beds. Because of these threats, bonefish have been classified as near threatened by the International Union for the Conservation of Nature. In the late 1990s, anglers and fishing guides noticed that the bonefish population in the Florida Keys was declining, so they banded together to create Bonefish and Tarpon Trust to understand and restore the fishery. Recent interviews with fishing guides and anglers underscored those concerns by showing a decline in the number of chances at bonefish during a day of fishing since the 1970s. Research since BTT's creation has greatly improved our understanding of bonefish biology. We have many of the dots figured out, but we can't yet draw the lines that show us the full picture of how to conserve and restore the bonefish fishery. The Bonefish Reproduction Research Project is aiming to fill important knowledge gaps. Exactly where do bonefish spawn? We have documented pre-spawning schools inshore, and we know they spawn offshore, but where and how deep? It's a big ocean. We also need to learn about their physiology, so we can understand what makes them tick. How do so many bonefish, scattered over such a large area, know it's time to migrate to spawn at the same time? After hatching from the eggs, what is the behavior of bonefish larvae? And how does their behavior influence the way they are transported in ocean currents? We are also raising bonefish in captivity to help us understand their biology. We want to complete the drawing so we can devise better conservation measures to protect the fishery for future generations. Although scientists have been making steady progress increasing our knowledge of bonefish, new sources of support have recently increased the rate of research. In 2018, we had the good fortune to be approached by the Fisheries Research Foundation with an offer to use the motor yacht Albola to assist with this important research. Because of the crew and fleet of boats, the Albola enables us to spend a week or more conducting research 24 hours a day in remote locations. We use shallow water boats to survey the flats in search of bonefish schools. When schools are spotted, we use our seine net to encircle the school. Once the fish are in the net, we measure and tag the fish and record the date and location where they were tagged. When a fishing guide or fisherman catches a tagged bonefish, they report the information to our website. So far, nearly all of the bonefish recaptured were within 1 mile of where they were tagged. We also determine the sex of the fish and whether they are getting ready to spawn. Soft pressure on the abdomen can induce a male to emit sperm. For females, we use a process called cannulation. If we get eggs, they are saved in a vial and put on ice for later analysis. We also take blood samples from females. More on this later. Although bonefish have small home ranges for much of the year, our tagging information has revealed that they migrate long distances to spawning locations. These migrations can be more than 70 miles each way. The Albola has been especially helpful as we study the bonefish spawning process. We anchor the yacht near a bonefish pre-spawning site where we study schools that can number more than 10,000 bonefish. We do this by quickly catching fish with hook and line. We then surgically implant a tracking tag in the bonefish while still on the deck of the flats boat. This allows us to quickly release the fish back into the school so it won't be eaten by sharks. Just before dusk, we switch to a larger boat. From this boat, we use a hydrophone to listen for sounds that bonefish might make as they get ready to spawn. We also pilot an underwater drone to observe bonefish behavior. This is an aerial view of the underwater drone next to the bonefish school. And here's the view from the underwater drone. At dusk, the bonefish begin to move offshore to spawn. We use special equipment called a VR100 that allows us to hear the pings from the tracking tags. In the foreground, you can see the blue handle on the pole that holds the hydrophone that listens for the tag underwater. The tracking continues through the night. The tags also record the depth and water temperature where the fish are swimming. Even as we track the fish using the VR100, we are also able to see the school on the sonar. This helps to verify the tag readings. You can hear the scientists call out the readings from the VR100 as we watch the school on the sonar.
Okay, we're at 127.6 meters, 26 degrees Celsius. Now at 128.2, 129, 26 degrees Celsius.
On a recent research cruise, we tracked the bonefish swimming to 137 meters, or 450 feet deep. This computer animation shows the dive. The brown at the top of the video depicts the island's shoreline. The blue background is the steep wall dropping to thousands of feet deep. This is the first time that a fish that lives in shallow water has ever been recorded diving to such depths to spawn. This is truly an amazing discovery. Back in the lab, we have a lot of work to analyze the samples we collected from the field. Remember that we collected blood from female bonefish. We use the blood to examine how the concentrations of bonefish hormones change over time. After collecting the blood, we place the tubes in a centrifuge. A centrifuge is a machine that spins the vials at a very fast speed and separates the plasma from the rest of the blood. The plasma is clear and contains hormones. We use a pipette to extract the plasma from the rest of the blood sample. We then pipette the plasma into wells in a microplate tray. We place the tray into a microplate reader to determine the concentrations of hormones. We are examining two hormones, estradiol and testosterone. This graph shows the difference between lower hormone levels on the flats compared to higher levels in bonefish in the pre-spawn school. This will help us determine the readiness of bonefish to spawn in the wild and in captivity. Remember that we also collected eggs from females on the flats. After we collected the eggs, we put them in a vial and a cooler on ice and transferred them to a freezer. The next step is to analyze the contents of the eggs. To do this, we first grind the eggs using a special machine called the bead ruptor. We put the eggs in a vial with small beads and a special solution. Then the machine shakes the vials. This breaks up the eggs so we can examine their contents. Our goal is to see how many lipids, or fats, are in the eggs. The more lipids, the healthier the eggs, and the greater the chance the baby bonefish that hatch will survive. After breaking up the eggs, we put them in new vials and then put them into the centrifuge. This separates the lipids from everything else. The next step is to use a special process to concentrate the lipids even more, using nitrogen to help evaporate the liquid. The remaining concentrated material is carefully put into a tray that contains a series of tubes. These are then analyzed by an iatroscan machine, which gives us a readout of the results. The results show the percentage of lipids in the eggs for each female bonefish that we sampled. In general, healthier females will have more lipids in their eggs, which leads to a healthy next generation. In human terms, think of it as healthy females don't need supplements. Now that we know where bonefish gather to spawn, and we know their biology, we're using the albula as a platform to take the next steps. We want to spawn bonefish in captivity so we can study the growth of young bonefish. Remember when we caught the bonefish from the pre-spawning school, tagged them, and put them back into the school? We also kept some of those fish and took them back to the albula. Once the fish were in the tanks, we checked to see if they were ready to spawn. We cannulated the females just like we did on the flats. If the eggs looked close to spawning, we would then give them a hormone injection to help the process along. A few hours later, the eggs were closer to being ready. We then strip spawned the female by applying pressure to her abdomen. We fertilized eggs using a spawning-ready male. We added water and stirred the eggs and sperm to make sure that the eggs were fertilized. And we placed the fertilized eggs in special tanks that slowly moved the water in a circular pattern. These are the first ever images taken of bonefish embryo development. You can see the embryo develop every few hours with each image. After 24 hours, just before hatching, the bonefish embryos release enzymes that start to weaken the egg wall. Then the embryos begin to shake to break out of the egg. This is the first time anyone has documented bonefish embryos hatching. Once they hatch, they don't look anything like a bonefish. Instead, they look like a clear eel. This is called a leptocephalus. This video shows hundreds of newly hatched bonefish at the water surface in one of the hatching tanks. Their development is amazing to watch, from small larvae with no eyes and barely able to swim, to actively swimming and searching for food. We have been able to raise larvae for 8 days, which is the first time anyone has been able to do this. Overall, it takes bonefish about 2 months for the larvae to completely develop, like this one, which was filmed in the wild. Soon after, the larvae change shape into a miniature version of their parents. As we learn more about bonefish larvae, we will be better equipped to understand how different populations are connected. Now that we know what makes bonefish tick, what healthy bonefish reproduction looks like, our next step is to repeat this in captivity. Can we get captive bonefish to spawn and produce healthy offspring? To address this next challenge, we worked with fishermen in the Florida Keys to catch bonefish from the flats. We transported them to the dock and then transferred them to large tanks. We loaded the tanks into trucks and braved the traffic of Miami to get the fish to Harbor Branch. After giving them a few months to get comfortable in their new home, we began the spawning process. First, we lower the water level in the tank to make it easier to catch them. Once a fish is scooped up, it's scanned. Each fish has an internal electronic identification tag. Matching the tag number on the datasheet tells us if the fish is a male or female. We then place the fish in a smaller tank with an additive to make it relax. This makes the process less stressful for the fish. We then cannulate the females to check on the status of their eggs. We place the eggs on a microscope slide and use a digital microscope to measure egg size. If the eggs are still too small for spawning, she gets a hormone injection to help egg development. She is then transferred to a recovery tank. She'll be checked again next month, along with the others that are not ready to spawn. We also collect eggs to analyze the lipid content to compare with wild fish. If a female does have larger eggs and we find a male that has sperm, we place these fish in the honeymoon suite to see if this gets them into the spawning mood. The honeymoon suites are smaller tanks with controlled lighting and temperature removed from the rest of the fish. So far, we have not had our captive bonefish spawn, but we are getting closer with each attempt. Once we do, we'll have the ability to raise and spawn bonefish in captivity. This might one day be useful for helping a population recover from a tragic event, like the strong cold front in 2010. Our work has already helped to create new national parks in the Bahamas, and we're discovering new spawning sites each year, from the Bahamas to Cuba and Belize. Now that we know what healthy bonefish eggs look like, we have a realistic target for our captive fish, and we have better knowledge for evaluating wild fish populations. Now that we know that bonefish spawn more than 200 feet deep, we can revise our estimates of where these larvae are transported by ocean currents. This will help us create better strategies for regional management. We haven't quite completed the picture, but the future holds great promise. It's amazing how much we've learned in just a few years.